Hello. Today I'd like to talk to you about some of the work that we're doing in our studies on the diversification of Hawaiian Cyrtandra. So this was work that was published in 2019 in a collaboration with two previous graduate students in my lab, Joseph Kleinkoff and Wade Roberts, and Warren Wagner. And we have to also thank many other people for help with some of the techniques and helping to provide specimens and feedback on our approach. So Hawaiian Cyrtandra has a very complicated taxonomic history. It's been treated at least 6 times. There's a large amount of morphological diversity, and a lot of this variation is quantitative shape variation, and this makes it very difficult sometimes to make decisions about how to circumscribe that variation into species. And we really want to try to better understand how these complicated patterns of morphological variation really contribute to kind of the lineage perspective on diversification in Hawaiian Cyrtandra. Part of the complexity to Cyrtandra is in its taxonomic history. Harold St. John recognized more than 400 species of Cyrtandra just from Oahu. And so he was parsing morphological variation very finely. And subsequent workers, Warren Wagner and colleagues, have recognized about 60 species and about 89 putative hybrid combinations. So this is a very different perspective on morphological variation than St. John. We have more morphological variation within and among populations, and it's recognizing that hybridization is likely contributing to some of the morphological variability. So these 60 or so species, most of them are single island endemics, and we'll kind of address some questions of some of the non-single island endemics as we look at some of our results. We think hybridization is common when Cyrtandra species are sympatric, and we have a number of putative hybrid combinations on all of the islands in the Hawaiian island chain. Previous phylogenetic studies have been very inconclusive about relationships. We've struggled to find much molecular variation, and the different phylogenetic hypotheses from different genes often conflicted in their putative relationships. So the outstanding questions that we are focused on here are really, how do we reconcile the genetic and morphological diversity? And can we determine species relationships, infer timing, magnitude, direction of gene flow, understand better the contributions of hybridization, and better understand the evolution of character traits in Hawaiian Cyrtandra. So we're using a phylogenomic approach, which we had a great introduction to in the workshop last week. For us, we are using the hybseq approach. So we've targeted a large number of low copy number nuclear loci, and this allows us to more kind of explicitly test for hybridization with this data. So again, the general idea here is that we are sequencing lots of genes, and we are making gene trees from those genes to build species trees. And we can look at this kind of diversity of gene trees and how they correspond to the species tree to try to test different hypotheses, including incomplete lineage sorting and hybridization. So our sampling for this study included samples from across the Hawaiian Islands. In some cases, we included multiple samples within species. We included a couple of things that are putative hybrids to test our analytical approaches. and we include a couple of things that are putatively undescribed species. For most of the analyses, we are looking at Maui Nui as a whole because we have to have more than 4 samples within an island group to do some of our analyses. So we were able to get a lot of data. We were going after 570 gene exons, and our final data set included 569 of them. And when we look at the aligned bases across the data matrix, we have more than 460,000 aligned base pairs. We also have a lot of variation that seems to be informative about phylogenetic relationships. So I'm not going to talk here about concatenated analyses. You can see more details about that in the paper, but our species tree analyses give us a very robust phylogeny of Hawaiian Cyrtandra given these samples. We see evidence of the progression rule moving from Kauai to Oahu and Maui Nui with some possible back dispersals. We have strong support for much of the tree. We do see some patterns that are curious that we want to explore more, including the fact that Cyrtandra hawaiiensis comes out in multiple locations in the tree. It is one of the few multi-island species recognized in Hawaii. And we also see multiple samples of grayeye and Monroe coming out in different locations in the phylogenetic hypothesis. So to test for the influence of hybridization in comparison with incomplete lineage sorting, we use a number of different analytical approaches. I can't go into all the detail of these approaches here, but I'd be happy to talk about them more in the discussion. SNAC and PhyloNet are explicitly phylogenetic approaches. The ABABABA test is a more kind of population genetic metric. I'm not going to talk much about it here, but generally the 3 methods were very similar in the results that we were able to discern. We did the hybridization incomplete lineage sorting test by island. We only had 4 samples from Hawaii, and so we were not able to test for hybridization there. So when we look at Kauai, all of the analyses converged on the same result, that we had 1 hybridization event. And that hybridization event was associated with a sample that we had a priori determined to be a hybrid in the field between Longifolia and Kauaiensis. Now, interestingly, the SNAC analysis would suggest that it's not an F1 hybrid, that it is at least a F2 backcross to Longifolia, given the genetic contribution estimates that we see from that analysis. So this was a good test to see whether or not something that we were fairly positive was a hybrid, would it actually be determined as such using this analytical approach. Oahu is obviously a much more complicated situation. We have many more species. We have many more putative hybrids. With our sampling, we ended up with some analyses suggesting 0 hybrids, others suggesting 2 or 3. So we'll look at the 0 versus 2 comparison. Now, even if there's not inferred hybridization, we have questions about what Hawaiiensis is. It comes out in 2 different locations. And when we look at possible hybrid edges, one of the interesting things about this pattern is that it would suggest that we have historical hybridization events that have then led to diversification of clades. And we see these kind of nested putative hybridization events. Now, one of these putative hybridization events involves Calantha, which is a very rare species that has major conservation concern. Interestingly, this analysis would suggest that if there was integration, it was between Hawaiiensis and grandiflora rather than between Hawaiiensis and Calpidicarpa, as has often been presumed. In Maui Nui, we hypothesize either 0 or 2 hybridization events given our samples. One of them looks like a F1 hybrid. It was actually a sample that was not a priori determined to be a hybrid. It was identified as Hawaiiensis. And interestingly, a putative hybrid between Hawaiiensis and Grayana is not hypothesized to be a hybrid here. As with Oahu, we see one hybrid edge actually leading to a diversification event, Grayana and Macrocalyx. We also see that Monroe is not coming out monophyletic. The 2 Monroe samples, each from different islands in Maui Nui, would suggest that the morphological variation that we see between these samples might need to be reevaluated. There might be more species that should be recognized here than we currently recognize. So in summary, these approaches, I think, provide a very powerful set of tools for understanding species relationships in gene flow in Hawaiian Cyrtandra. We have large amounts of incomplete lineage sorting, but this preliminary data would suggest that we really can tease apart these influences between ILS and hybridization. The overall pattern, though, is one that we would expect to see in Hawaii with the progression rule in evidence from our phylogenetic hypothesis. So where we go from here is we really need to have dense population sampling of all of the species on each island to really better test where hybridization events are occurring, being able to actually include putative hybrids and their parents to see whether or not those morphological variants really are hybrids, or if there is more morphological variation within some of these species than we have previously appreciated. And then this approach will also allow us to answer broader questions about the diversification of Cyrtandra in Hawaii and really try to get at some kind of fundamental questions of evolution and diversification using Cyrtandra to answer those questions. Thank you.